Stem cells are an important tool for scientists to study how humans develop and how this goes wrong in disease. Here in the Medical Research Council Protein Phosphorylation and Ubiquitylation Unit, or MRC PPU for short, we use these cells to try and find the causes of human developmental disorders, such as intellectual disability. Intellectual disability is characterised by impaired learning and social functioning, which affects an estimated 1 to 3% of the world's population. However, the causes of intellectual disability remain mostly unknown. Let's visit our tissue culture suite and see how we use stem cells in our experiments. First, we must ensure that stem cells are in the right environment to grow healthily. Like ourselves, they need warmth, air, and nutrients to survive. Warmth and air are provided by cell incubators and nutrients by a liquid soup. We regularly check that the stem cells are healthy by using a microscope. Then, before starting an experiment, we first count the cells to make sure we have enough. We do this by washing off the nutrient soup they grow in and add an enzyme to lift the cells off the dish. The cells are counted using an automatic counter, and we take the number of cells we need to start our experiments, which is normally at least 1 million. Cells are then returned to the incubator to grow. The stem cells we study are from the embryo, which means that they have the ability to become any cell type in the body. We can treat stem cells with various chemicals and factors to mimic the environment within the body. This allows them to start to develop into specialised cell types that make up our tissues. For example, neurons from the brain or cardiac muscle cells from the heart. We can then use these cells to study how tissue development is controlled and how this might go wrong in people with developmental disorders. To unravel the communication systems that control tissue development, we must break open the cells to find out what is happening inside. We can then separate and collect the cellular instruction manual, DNA and RNA, and molecular machinery, proteins, from inside the cells. These parts can then be analysed using different experimental techniques. This reveals how tissues develop, how this goes wrong in people with developmental disorders, and how we might better treat these diseases.